My name is Taylor, and I'm one of the pediatric genetic counselors here at Sanford Health. And today I'm going to be talking about whole exome and whole genome sequencing. So this presentation is titled Whole Genome and Whole Exome Sequencing, Is It for Everyone? So like I said, kind of an overview of the talk today. I'll talk about what these two technologies are, what whole genome and whole exome sequencing are. I'll talk a little bit about how they're done and how we use them in clinic with our patients here at Sanford, and when we consider these two tests, what are some of the benefits and limitations of them, what are the possible results that a patient could get if they have one of these tests done, and then also I'll talk a little bit about is this for everyone, and how else could these technologies be used. So to start off, what is whole exome sequencing and what is whole genome sequencing? To kind of start off, we'll go back in a review of genetics. So every single cell in our body is made up of, every single cell in our body has a copy of our DNA. And our DNA is, we get it from our parents. Half comes from mom, half comes from dad. One of the terms that we use when we talk about DNA is our chromosomes. And what a chromosome is it's a long strand of that DNA all packed up together. And each chromosome contains hundreds of different genes on it. And our genes, what those are basically recipes or instruction manuals for the different things that our body needs to do to grow and to function and to develop properly. Our genes, they code for different proteins. And our genes our DNA sequence is made up of a ton of different letters, or not a ton, there's just 4 specific letters that make up the sequence of the alphabet of our DNA. So when we think about kind of what those genes do is they code for proteins, and our proteins, like I said, they help our body to work and to develop and to function properly. So thinking about our whole exome and whole genome sequencing, those tests, we'll start off talking what is the genome. The genome is the 3 billion base pairs or 3 billion letters that make up our DNA. So it's that entire strand of DNA, every single letter that makes it up. That would be our genome. And it's about 3 billion letters. The exome is just a portion of the genome. It's about 1 to 3% of the total genome. And the exome, what it is it's the exons. And the exons are the portions of the DNA, the portions of that DNA sequence that are the ones that encode the proteins, the portions that are the genes. So not all of our DNA is all genes. There's kind of portions in between the genes, and even within the genes that we don't really know as much about the function of those and what the function of those portions of our genome is. So the exome is just those protein coding portions of the genome. So one way to think about the difference between the exome and the genome is if we were to think about a football game or another sports game, the genome is like watching a football game from the beginning to the end, getting every single little detail, whereas the exome would be like reading about the highlights of that game the next day in the paper. So going back to whole exome sequencing and whole genome sequencing, what is the sequencing part? So sequencing is basically going through, getting all the reading through those letters of the exome and genome, those letters of the DNA sequence. So for whole genome sequencing, it's reading through and getting that sequence for all 3 billion base pairs, 3 billion letters. For the exome, it's reading through those exons and getting the sequence for those letters. So thinking back about kind of what our genes do, we know that if there's changes or mutations in the sequence of the gene, that can affect how that gene functions, or it can make that gene not produce the proteins that it's supposed to be producing, or can make that protein that's produced be abnormal and not work properly, which can lead to different health problems. So, we refer to these as mutations or changes or variants, lots of different words that we can call them, but these are basically changes in the sequence of that DNA that affect the way a gene would work. So, in a nutshell, what are whole exome and whole genome sequencing? These are genetic tests that involve sequencing the exome or the genome. So how exactly are these tests done? So typically we obtain a blood sample from a patient if they're going to have this testing done. Sometimes we can use a saliva sample or another tissue sample, since most of the cells of our body do contain our DNA. So once we get that sample, we're going to isolate or extract the DNA from it. And then from there, what's done is that we sequence that DNA. So basically do a spell check of it, read if it's genome sequencing, read out all 3 billion pairs of that sequence. If it's exome sequencing, then we're reading out the exome, getting the sequence for that. So when we do that, when we sequence it, after we get our full sequence of all the letters, then we compare it to a reference sequence, kind of what we would expect to see in the typical human exome or genome. And typically there's going to be hundreds of thousands of variants that are identified, changes from that reference sequence in that individual sequence that we find. And those changes, most of them are just benign changes that don't cause any health problems at all, and are just things that make us unique. Maybe it's something that makes our hair color brown versus blonde, or our eyes blue versus green, or makes us taller or shorter than someone else. Most of them aren't disease-causing pathogenic mutations. But some of them, kind of the point of the testing most of the time is to figure out if any of them are a disease-causing or pathogenic test. So the next part of the testing is the analysis, and that's a really important part, because what that is trying to determine if any of those thousands of changes that are identified are the disease-causing or pathogenic changes. So that's done by someone, and they'll go through and look at kind of sort those variants, figure out which ones might be the most likely to be disease-causing ones, and then from there determine if any of them are ones that they do think is the cause of that patient's symptoms or health concerns. And then the last is that we'll get a report with those results. So that's kind of an overview of whole exome and genome sequencing. Now we'll talk a little bit about who should get this testing done. So in a clinical genetics setting, we definitely offer this testing for some of our patients. When we're thinking about patients that this testing would be a good option for, it's typically individuals with medical concerns that could be due to an underlying genetic cause. So these are people who we think might have some type of genetic condition. But it's not everyone. We usually think about kind of other factors we'd want to consider as well. So the ones who should have whole exome sequencing, or what patients that we might consider it for, are people who are thought to have a genetic condition, but maybe previous testing or their previous genetic workup hasn't been able to identify a genetic cause. Whole exome and whole genome sequencing usually isn't the first test we jump to. Typically, we'll do some other types of genetic testing first to kind of rule other things out. Whole exome and whole genome sequencing are pretty broad, comprehensive tests. They're kind of the most broad tests we have right now available in genetics. So typically, we'll kind of focus more on more specific tests first for individuals. But some patients, whole genome or whole exome sequencing is the first test that we would want to jump to. In these cases, for patients when there's multiple different genetic conditions that we're considering or that the doctor's considering for them, and maybe it would be more cost-effective or quicker to just start with this broad test instead of doing a bunch of different tests first, if we're not really quite sure where to start. So other times when we would consider whole exome sequencing is, like I said, if there's a long list of differential diagnoses. So if there's a patient where they might have this condition or this one or this one or this one, if there's a ton of different conditions we're considering, then rather than testing, for each specific condition, we might just start with this whole test, since it's going to be able to look at all the genes at the same time. Also might use it for unclear, atypical patients where we think there's something genetic going on, but we really have no idea what it is. So maybe this is a good place to start because we don't know where else to start. Also for patients who have kind of been on a diagnostic odyssey, this can be a good test. If they've had a ton of different tests and seen lots of specialists and they just haven't been able to figure out a cause for what's going on, whole exome or whole genome sequencing might be a good option for them. It's also a good option if there's limited or no clinical testing available for this suspected condition that a patient is suspected to have. So this is a test, since we're looking at all the different genes, we would technically be able to look for things even if there's not a test available for that gene we're suspecting the patient might have a change in. Also, if we're suspecting a novel or new condition that hasn't been reported very much, this could be a good option. Or if we're suspecting a condition with high locus heterogeneity. So what that means is that there's changes that in many different genes that have been reported to cause the suspected condition. So if that condition has gene X and gene Y and gene Z, all that are associated with that condition, then this could be a good test to kind of get all of those genes at once. And then also if the clinical features or the symptoms that the patient is having are suggestive of multiple different conditions with overlapping kind of features, and we're not really sure how to tease out which one would be the most likely condition for that patient. So lots of different times when we might consider this test, but it's not the best option for all patients. So when would it not be the best option for a patient? If the individual is highly suspected to have a specific genetic condition that has a testing option available for that specific condition. So for example, if we suspect that a child has Down syndrome, we're going to test specifically for Down syndrome. We're not going to do whole exome sequencing for that individual. Another kind of time when we might not want to consider whole exome sequencing is if an individual is suspected to have a genetic condition caused by a type of change that isn't detected by whole exome sequencing. So as I said earlier, it's a pretty broad test and looks and sequences all the genes, but it doesn't detect all types of genetic changes or variations. So it wouldn't pick up, large deletions or things, trinucleotide repeats, different things that aren't sequence changes. And then another thing to consider is the cost. It's a pretty expensive test, so for individuals whose insurance might not cover it, or if the family isn't able to pay for it or doesn't want to pay for it, it might not be the best option for them. There might be more cost-effective approaches to consider for that family. So some of the limitations of the testing and kind of other things to consider about whole exome and whole genome sequencing are first the technological limitations. So like I said, it's a pretty broad test, but it's not perfect, and definitely we have incomplete coverage with the test. And what that means is that there's areas of the genome or the exome that we're not able to get a sequence for when we do this test. So we don't get a sequence of 100% of the genome and 100% of the exome. Our coverage is less than that for these tests when we do it right now. So we might miss changes in the areas that we don't have complete, or areas that we have poor coverage for might be missed. Also, there's a reduced ability to pick up certain types of genetic variations, like I said earlier. So large insertions or deletions or trinucleotide repeats and copy number variants, those types of genetic changes or variations are things that we might not be able to detect by this technology. So if we're suspecting a condition that's caused by one of those types of changes, then we'd need to consider other testing options. And then there's the inability to analyze all the variants identified. So when we do one of these tests, we're identifying thousands of different variants, and we're not able to look through each variant, specifically and determine and thoroughly research it. They have to prioritize and kind of filter out which variants are going to be the ones we focus on and really research to see if that could be the cause for the patient's disease. So that's another way we might miss some variants. And lastly, kind of the incomplete knowledge of genetic causes of disease. So our knowledge of genetics and the causes of different genetic diseases is constantly improving and changing. And there's always more research being done. So with this testing, we're limited by our current knowledge. So if a genetic condition isn't identified today, this testing isn't going to be able to detect it. But maybe we'll find some new genetic condition in 5 years, and then we would be able to detect it by this then. Lastly, another kind of thing to consider, as I mentioned earlier, is the cost. So whole exome and whole genome sequencing are pretty expensive tests. So that's definitely something that has to be taken into account. And then the possible results are one more thing that we always need to consider. So we'll go into talk a little bit more about those in more detail. So for whole exome and whole genome sequencing, the possible results that we can get are results related to the reason for testing. So when we do this test on patients in a clinical setting, we're doing the test to try to figure out what the cause is for their health concerns that they have, the symptoms that they're experiencing. So if it's a child with seizures and developmental delays, we're trying to figure out, is there a genetic cause for this child's seizures and developmental delays? So those results related to the reason for testing would be the results that would tell us if there is an identified genetic cause for those seizures and developmental delays. But there's also other possible results that we can get from this testing. So we'll talk a little bit about both of those things, starting with the results related to the reason for testing. So the possible results we could get are positive, meaning that a pathogenic variant or change is identified that is associated with the individual's symptoms. This would give that individual an answer and may give them a specific diagnosis of a specific genetic condition. On the other end, we could also get a negative result. What that means is that there were no variants or changes identified that were thought to be associated with that individual's symptoms and features. The important thing to remember is that this doesn't rule out a genetic cause for their symptoms and features. This testing doesn't detect all types of genetic variations, and it doesn't test for all conditions, and there's definitely limits to our current knowledge today about genetics and the cause of genetic diseases. The last possible result we could get related to the reason for testing is an uncertain result. So in this case, it could be that there's a change that was identified, but we're unsure of the significance of that change. We don't know if it's just one of those benign changes that makes that person unique, or if it's an actual pathogenic disease-causing change. Sometimes we can also find changes in genes that are kind of genes that we don't know very much about yet. We don't know if that gene is associated with disease, but we think maybe it could be. So those are uncertain results. In these cases, it wouldn't give a family or a patient a definitive answer or genetic cause for the symptoms that that individual is experiencing, but would be something to take note of and possibly reinvestigate in the future with hopes of further research or better understanding of those genetic causes or what was identified. Then we move into the other possible results for this testing. So one of the possible results we can get when we're doing this testing is knowledge about unexpected family relationships, such as non-paternity or family relatedness. So most of the time when we do this testing, we'll get samples from a patient as well as their parents. And if we're getting samples from parents, then if dad is the individual who's reported as dad isn't actually the biological dad, that's something that might be discovered by this test. We may also be able to determine if an individual's parents are related or are cousins or second cousins or something like that is something that might be able to be picked up by this testing. Then the other category of other possible results is kind of secondary findings. And these include conditions of predisposition for genetic conditions. So these are genetic conditions that typically aren't the reason that we're doing the testing for. They're mostly hereditary cancer syndromes and hereditary heart conditions, including connective tissue disorders and cardiomyopathies and arrhythmias. So these are, there's a list of 56 different genes that are reported by the American College of Medical Genetics, and I believe they're updating that list soon. But these are genes that are associated with these hereditary cancer syndromes and hereditary heart conditions that the American College of Medical Genetics recommends that if someone's having testing done that looks at these genes, they report out any pathogenic changes in these genes. The reason that they recommend that is because these are genes that are associated with genetic conditions that have a treatment or management plan available. So if someone, for instance, has a change in their BRCA1 or BRCA2 gene, that's associated with an increased risk for breast cancer and ovarian cancer and some other types of cancer. If someone has one of those changes, then there's specific management and guidelines that they can follow to help look for and screen for those cancers and reduce their risk of developing those cancers. Depending on where the testing is done, there are some other conditions that might be available to be reported as well. There's a number of different labs that offer whole exome and whole genome sequencing, and they each kind of have different secondary findings that they might report. Some will report carrier status, and some will report pharmacogenetic information as well. Up here is a list of the 56 genes that are recommended to be reported by the American College of Medical Genetics, including those hereditary cancer syndromes and hereditary heart conditions mostly. So going back to the results related to the reason for testing. For each patient, it's going to vary on what the most likely result is that we're going to get for a patient, but how often do we get a positive result though? And like I said, it really varies by the patient and why they're having the testing done, what their specific family history is, and what their features are. But this table here shows a little bit about from one of the testing labs, what they've seen with the percentage of how often they're able to get a positive result. And as you can see, it kind of varies based on the main presenting features of that patient. So for an individual who has skin differences as their major feature, they have a pretty high chance, a 46% chance of getting a positive result. Whereas someone with endocrine problems as their main reason for having the testing done tends to have the lowest chance of getting a positive result. So thinking about this testing, is it for everyone? In a clinical setting, I would say no, this test isn't for everyone. As I already mentioned, it's not going to test for everything. It's going to not detect certain types of genetic variations. It does have a pretty high cost, so it's not always something that we're going to want to jump to this as the first option if there's more cost-effective options for a family that could get them the answers that they need. There's also a kind of low chance of getting a positive result. Like you saw on that last slide, not everyone is going to get a positive result. So there might be better testing options available for someone. And then there's always the possibility that there's other more appropriate tests available. And then lastly, the technological limitations of this test and our incomplete knowledge of the genetics of disease are kind of another reason why this might not be the best test for everyone. So what about healthy individuals who, we're not seeing in the clinic for a reason to try and figure out what the cause of their genetic symptoms are? Healthy asymptomatic individuals. Is this a good option for them? And I think that's kind of an uncertain thing, but there's definitely some things to consider with it. Some clinics and companies are offering or aiming to offer this technology to healthy individuals without a medical indication for it. And I think, like I said, there's a lot of things to consider when we're thinking about doing this types of testing for healthy individuals. So, it's a complex test, as you've learned from listening to me talk for the last little while here. So I would definitely encourage anyone considering to do this test to meet with a genetics professional to make sure they really understand it and are able to get all the questions they have answered. Also, the other thing to consider is that there's implications for other family members. Whenever we test and look at our DNA, we get half of our DNA from mom, half from dad. So our family members will have some of the same things to us. So if we're looking at our DNA and figuring things out from there, it's going to have implications for other family members as well. And then there's the kind of idea that more isn't always better. Having more information is definitely useful in some cases and for some people, but it might not be the best. More information about your genetics isn't always the best option for everyone. Another thing to consider is what information is actually going to be reported. So when I was talking with you guys in our clinical setting about what the possible results are, the main results that we report are related to the reason we're doing the testing, what that patient's symptoms are. So if someone's healthy and doesn't have any symptoms, what is going to be reported? What results are they going to get? Is it going to be just those 56 genes recommended by the American College of Medical Genetics? Are they going to report carrier status? Are they going to report other things? Are they going to report every single possible genetic condition? That would definitely be an important thing to consider if this testing is going to be offered to healthy individuals. And then also just the fact that, it may not provide information that is useful to everyone or wanted for everyone. We know that many individuals, even if they're at risk for developing an adult onset condition and have a family history of it, they might not want to have testing done for it. They might decide they'd rather just kind of wait and see if they develop it. So more genetic testing isn't always the right choice for everyone. In conclusion, I'd say these are definitely great technologies, and our use of them in the field of genetics is only going to grow in the future. But I definitely encourage you guys to talk with a genetics professional if you do have more questions about these technologies. So thank you so much for listening and have a great day.